right, in chapter 7, we're going to look at the control of microbial growth. In chapter 6, we talked about the requirements of microbial growth. Now we're going to look at how we can control it. So some definitions that we need to know are bactericidal versus bacteriostatic. Anytime you add "cide" or "cidal" to the end, this is a suffix that indicates destruction of a type of microbe. Okay, so when we say bactericide, it's going to kill bacteria, fungicide kills fungus, and so on. This does not necessarily take care of the endospores though. On the other hand, when you add stasis or static to the end, this is a suffix that indicates inhibition, but not complete destruction of a type of microbe. So it prevents it from replicating and growing, but it does not kill them. All right, this would be bacteriostatic, meaning that it's going to inhibit bacterial growth. Fungal static is going to inhibit fungal growth. Virostatic are inhibiting viruses. All right, and so when we look at this, there is a difference between it being cidal versus static. All right, some other things we want to look at are some terms like sterilization. Sterilization is the destruction or removal of all forms of microbial life. Now, this does include endospores, okay? But with the possible exception of purons or prions. Because of this, they're harder to potentially get. Sterilization may not take care of them, but it does take care of endospores. If we look at disinfection, this is going to be the destruction of vegetative pathogens. It's not going to take care of endospores, but it will take care of the vegetative pathogens. We also have antisepsis. This is destruction of vegetative pathogens, but this is going to be used on living tissue. All right, so there is a difference between a disinfectant versus an antiseptic. So an antiseptic, guys, is mild enough to be able to be used on your skin. Some examples are hydrogen peroxide, mercurochrome, tincture of iodine, and 70% isopropanol alcohol. On the other hand, a disinfectant is going to be used on inanimate objects. Examples of this are going to be bleach, heat. We'll also look a little bit at commercial sterilization as well as degerming and sanitation, which are all just a little bit different. Some of these types of microbial growth control are better than others. There are some factors that influence the effectiveness of an antimicrobial, something that works against microbes. First, we have to know the number of microbes. The greater number of microbes, we have to increase the killing time. And so because of that, you'll have to increase the amount of time that chemical or that heat or whatever it is that you're using is in contact with those microbes. Another thing are some environmental influences, the idea that it's in body fluids. If it's found in body fluids, you also need to increase the killing time. Time of the exposure, the longer time that you expose it to, the increase of its effectiveness. So if 10 minutes is good, 15 minutes might be better. Now, of course, there is a fine line where it's too much when we're talking about hurting the potential object that it's on, okay, or in. But when we're looking at this, though, increased time normally increases its effectiveness. Another thing is the characteristics of the microbe itself. And so when we look at these, microbes are kind of in a hierarchy. Some are more susceptible to antimicrobials than others. These include things like viruses with lipid envelopes, gram-positive bacteria, viruses without envelopes. These are all very susceptible to lots of types of antimicrobials, but others are more resistant, like endospores are more resistant as well as the purons. So there are some different actions that a microbial control agent can undergo. It's either going to affect protein synthesis, it's going to affect the DNA or the plasma membrane. So one thing is it could alter the membrane permeability. This could damage the membrane lipids or proteins, which diminishes the cell growth because it damages that ability of being able to regulate what moves in and out of the cell. Another thing we can damage is the proteins. It can denature the proteins by using heats or chemicals. And remember, when we denature a protein, it unfolds and it's no longer usable. Another thing we can do is actually damage the DNA and or RNA that's present in the cell. This is where you're damaging the nucleic acids. This hinders replication so that it can't make more of itself, and it also will slow down protein synthesis. So even though the proteins are working properly, if there's no DNA and RNA, then it's going to slow down that process as well. Some of these microbial control factors for growth are going to deal with the physical side, and some of them may be chemical methods. All right, what type of equipment or surface are you dealing with? This is going to help us decide whether we need to use something that's physical or chemical. Is it heat liable? Can you actually heat it? Some stuff cannot be heated up, like plastics. You can't heat that up too high, otherwise they'll start to melt. Is it disposable? Again, is it a type of plastic? Is it a metal? What type of microbe may need to be killed? All right, is it do you think gram positives are present? Viruses, purons, what do you think might be present there? Sterilization of media and pharmaceuticals are important. However, do they have a high protein base to them? Do those proteins important for them to be active? If it is, you cannot use a high heats because it denatures those proteins. And so again, you have to look at a lot of different factors to determine what kind of method you're going to use. There are established recommendations or protocols that are kind of put together in order for you to follow. Some examples of some physical methods include heat, filtration, even using cold, desiccation, osmotic pressure, and radiation. So we're going to take a look at some of these. All right, so if we're looking at heat first, these are again physical methods. When we're looking at heat, we can look at boiling. We can also look at autoclaving. We can look at pasteurization, and then also dry heat. Now, when we're looking at boiling or autoclaving, you'll notice that this does protein denaturation. So we see here that it's going to denature the proteins and make them where they cannot be utilized. Then we see here another thing that's important on this chart is the comment of that kills endospores. So if you look at boiling, it kills vegetative, bacterial, and fungal pathogens and almost all viruses within 10 minutes, but it's less effective on endospores. All right, so boiling might be good for some things, but not others. Autoclaving is a very effective method of sterilization at about 15 psi of pressure. This can bring the temperature up to 121 degrees Celsius. This will kill all vegetative cells and their endospores are killed in about 15 minutes. So if you're going to use one of these methods, autoclaving might be the better method versus the boiling. Pasteurization is a heat treatment for things like milk. It brings it 72 degrees Celsius for about 15 seconds and it kills all pathogens and most non-pathogens, those that are responsible for spoilage. Okay, but if you'll notice, it does not talk about endospores. We also see in this chart, it shows you preferred uses. When would we use these types of heat? Another thing deals with the dry heat. When we're looking at dry heat, you can do it directly over a flame. This would be like Bunsen burners or things like that, but normally it turns the contents to ashes. Also incineration also burns things to ashes. So if you're wanting to keep certain things, then we obviously don't want to use that. Another way is like with hot air sterilization, which is like an oven. This is very effective method of sterilization, but it requires temperatures of 170 degrees, but it's for a long time, okay, for 2 hours. Okay, and so we're looking at this, it could take a lot longer. All right, so the big thing is looking at what they can do, moist heat versus dry heat. All right, so what we see in lab is a lot of times we're going to be using autoclaving or direct flaming. The direct flaming is what you will have seen in lab where they will take the inoculating loop and things like that and put it directly into the flame. Or you'll take the slide when you want to heat fix it and put it directly towards the flame. On the other hand, autoclaving is how we get rid of a lot of our waste, making sure again that everything is killed before we throw it in the trash. In lab protocol, if you were in a face-to-face lab, if anything gets contaminated with the bacteria in lab, it has to be autoclaved, and this includes your clothes, your book, things like that. So it's very important that you are careful, and there's no guarantee it's going to come out the way it went in, especially if we're talking about a book, because we're using high pressure and also water in order to get that high temperature. So when we look at heat denatures proteins and kills endospores. Moist heat requires shorter time as compared to dry heat because water conducts heat better than the air does. All right, and so since the water can conduct that heat, it's going to have less time to be used. When we look at pasteurization, there's classic types of pasteurization. However, there's some more advanced types now that don't take as long. One is called the HTST FT or flash type of pasteurization. This is known as high temperature short time pasteurization. This decreases the bacteria, but it does so if you will immediately refrigerate afterwards. The other one is UHT, and this is ultra high temperature, or also known as ultra pasteurization. All right, and so this is using really high temperatures. The higher temperature, the less time is needed. Okay, and so if we can increase the temperature, we don't have to put it in there for as long. This does not achieve sterility when we're looking at pasteurization, and the whole purpose of pasteurization is to kill pathogens and to decrease the microbes associated with spoilage. It doesn't say it will get rid of them, but it does decrease them. That way the shelf life of it can be longer, like with milk. One of the main ways that we use heat in the lab is through a steam autoclave. The whole point of the steam autoclave is it allows you to use really high pressure and hotter temperatures to sustain the high heat. This is better than with boiling because it actually will keep it in a contained area. This is effective if material is to be sterilized, if it's not damaged due to high heat or moisture. All right, so like with glass, this one is an easy way to use that sterilization. The glass still comes out looking the way that it should, and so when we want to sterilize water or pipettes or things like that, this is used in laboratories and healthcare institutions to sterilize culture media, solutions, and instruments. And so they'll use a steam autoclave like in a surgical ward in order to kill the bacteria or any microbes that are on it, and then they repackage it and seal it, and then it has been sterilized. We also remember, if you remember from the chapter about the media, it's always important to sterilize it before you inoculate it to make sure nothing else is growing on it. So in the lab, if you're wanting to get rid of the biohazard waste, it's really important in order to do quality control to make sure that the stuff has died or is dead. One way you can do this is through strips. This one has a type of organism that is known embedded in the strip, and if it is killed, then you see that it gives you a color type of indication. Again, quality control is really important, especially before you throw something in the normal garbage, because you don't want it to potentially infect others. So when you're looking at steam autoclaves, they work on moist heat, pressure, and time. And so this particular diagram shows you kind of how it works. When we see this, if you're using a certain amount of pressure, it will raise it to a certain temperature, and then the time can be adjusted. And normally the standard is about 15 psi, which brings it to about 121 degrees Celsius, and this needs to run for at least 15 minutes. Now with dry heat, we use dry heat in the lab when we're flaming loops or forceps. We do this in order to sterilize them so that they can be used. We also see that you can use incinerators. These incinerators can sterilize. It destroys the material that is there. And so if you've ever used one of these little incinerators in a lab, if there's bacteria on your loop, you can actually hear it popping, okay, because it's killing it and then it gets removed. Again, hot air ovens could potentially be used, but the time has to be extended due to it being a dry heat. Another thing we can look at is filtration. Filtration, we must sterilize the equipment prior to doing the filtration. So all the equipment needs to make sure it's been sterilized. You can maybe use an autoclave, an oven, things like that. Passage of the liquid through membrane with pores too small for microbes to pass. All right, so the liquid can pass through, but the microbes cannot. We use a vacuum, which helps force the liquid to pass through the filter into the flask. It's used on heat-sensitive liquids. And so these are liquids that you can't autoclave or you can't use in like an oven or boil. So this is really important in separating bacteria from its suspending liquid. A note here is purons in a mycoplasma may be filterable. They are very small in size, and so we could potentially see that they are filterable. This is useful for sterilizing liquids, again, those especially who have enzymes or vaccines, to make sure that these things don't get destroyed by heat. So we have methods that deal with cold, things like refrigeration. We also have what we call desiccation, and we also have osmotic pressure on this particular chart. So this shows you the preferred uses. When we're talking about the cold side of it, we can use this for food, we can use it for drugs, and even culture preservation. On the other hand, desiccation and osmotic pressure is more for the food side, okay, in order to preserve food. One thing to note about refrigeration is that it is bacteriostatic. It is not bactericidal, but it does have a bacteriostatic effect. Desiccation and osmotic pressure, these guys are going to help disrupt either metabolism, like in desiccation, or osmotic pressure is going to cause plasmolysis. So let's look at the low temperature first. So with the low temperature, the effect varies per microbe. Some microbes, recall from previous chapters, they actually grow better in the cold temperature. Therefore, this would not be an effective way to control them. This is only bacteriostatic. It inhibits their metabolism. Refrigeration also reduces spoilage, but freezing may not kill all the organisms, but it could kill some of them. The idea with the freezing is that it can cause them to freeze up. That doesn't actually kill them, but when they start to thaw out, the water changes due to that thawing could potentially hinder the growth or even hurt the bacteria. The next one is the desiccation.